Hi friends, up till now we have been discussing various concepts of microbiology, biochemistry and metabolism in our channel. From now onwards we'll also be sharing few questions and their expected answers with you, which will help you to apply the knowledge in practical. So the question for today is the three-dimensional structure, that is the native conformation of a protein, is primarily determined by. Now there are various options for this, like they may tell you that may be determined by molecular chaperones, it may be determined by the amino acid sequence of that particular polypeptide, by the other proteins with which this protein is interacting, it may be determined by the non-covalent interactions in the proteins. We somehow tend to have more what you call as a tendency to answer like there are non-covalent interactions which will primarily determine it. No doubt they help and play a major role in maintaining this three-dimensional structure of the protein in a stable conformation. But the actual folding of the protein, it will be primarily determined by the amino acid sequence of that particular protein. So as will be the amino acid sequence, that will determine how that particular protein it will fold and which kind of shape that particular protein it will have. So the answer for this is the amino acid sequence of that particular protein, remember. The second question here says that how can a thermodynamically unfavorable reaction such as the synthesis of DNA or protein polymers, it occurs inside the cell. We know that this particular reaction is thermodynamically unfavorable because it has a positive value of delta G. Now we know that such reactions are unfavorable. Now various means by which it is achieved, one of the most common ways by which cell achieves this is by coupling this reaction to the hydrolysis of ATP. We know that ATP hydrolysis is a reaction which releases a huge amount of energy, which has a delta G0 prime of minus 7.3 kilocalories or minus 30.5 kilojoule per mole. So this energy or this particular reaction, when it is coupled to this reaction, you can have an overall negative value of delta G0 for this particular reaction. So a reaction which is thermodynamically unfavorable can be coupled to the hydrolysis of ATP so that the overall value of delta G is negative, and this value or this reaction which was earlier unfavorable can now be pushed in the forward direction. Thank you and stay with us for more such questions and answers.